Welcome, everyone. This is Jenny Glenn Gingery. I'm the Associate Director of Communications and Media Relations at the Endocrine Society. It's my pleasure to welcome you to our ENDO 2020 virtual news conference on endocrine disrupting chemicals, which, as you may know, are substances in our environment that mimic or block or otherwise interfere with the endocrine system. So we're pleased to have joining us today Dr. Barbara Cohn of the Public Health Institute in Berkeley, California, and Dr. Mohan Kumar of the University of Georgia. Over the next 20 minutes or so, each speaker is going to present their findings, and we'll have one Q&A session at the end of our time today. So everyone should be muted except for the presenters. We just do this to avoid any distracting noises in the background, and we would ask that you would type your questions into the chat window. Once both our speakers have finished their presentations, we are going to take questions at the end. Please note this event is being recorded. We're going to be showing it on our YouTube channel and will also be available in our online newsroom. So I'll be reading the questions out loud for the benefit of anyone who may be listening after the fact. So now I'd like to turn things over to our first presenter, Dr. Barbara Cohn.
Hello, good morning or good afternoon, depending on where you are. I am from the Child Health and Development Studies, which is a project of the Public Health Institute here in California, in Berkeley, California. And today I'm going to tell you a little bit about grandmother serum PFASs, and I'll tell you what those are in a minute, and their relationship to granddaughter's obesity. This work was to be presented at the ENDO 2020 meeting. It is not yet published. Next slide, please, Jenny. I have nothing to disclose. Next slide, please. So, and polyperfluoroalkyl substances, are called PFASs for short, are man-made chemicals, and they've been manufactured and used many places all over the world, surprisingly perhaps to you, since the 1940s. So two of them have been most extensively studied, PFOA and PFOS. PFOA most likely is what you know it might be in your Teflon pans. And both of these chemicals are very persistent. They stay in the environment and in the human body a long time. And there's some evidence that exposure to these chemicals can lead to adverse health effects. I direct you to this EPA website for some more information, basic information on these chemicals. Next, Jenny. So the question that people ask all the time is, where can we find these? And it turns out we can find them almost everywhere. In food that is packaged with PFAS-containing materials, that's been processed with equipment that use them, or grown in PFAS-contaminated soil or water. In commercial household products, long lists, stain-resistant, water-repellent fabrics, non-stick products, polishes, waxes, paints. You get the idea. Firefighting foams, which are a major source of groundwater contamination, which you may be hearing about recently in the press, and military bases where firefighting training occurs. The workplace, including production facilities that use PFAS. Drinking water, it's typically been pretty localized, but there's a very large map now that you can get on the Environmental Working Group website where you can see the places where we know there is contamination. And finally, inside living humans, fish, animals, where PFASs have the ability to build up and persist over time, and they seem to persist in organs like the liver. Next slide, please. So how can we possibly study parental PFASs and grandchildren's obesities? Well, we were able to do this because of an incredible study that I was 8 years old when it started, that was initiated in 1959. We enrolled 20,000 pregnancies by 1967, and it was a partnership with the Kaiser Foundation Health Plan in Oakland, California, and the University of California at Berkeley. This study is no longer in those two places. After 60 years, it now resides in a nonprofit called the Public Health Institute, where I'm its research director. These pictures are the children who were born into the study in the 60s as they were growing up. And these kids all gave permission for us to use their photos. And just to put this in perspective, these children are now between 50 and 60 years old. Next slide, please. So these are some of the things that we have done with the Child Health and Development Studies. And if you look at the far left, that the row called F0, those are the parents that we enrolled, the pregnancies we enrolled. And we've studied breast cancer in those women in relation to serum endocrine disrupting chemicals. That means chemicals that have the ability to alter endocrine function or response in humans. And we've published papers on breast cancer in the mothers. And we have published papers on breast cancer in the F1, those are the daughters, and also their breast density with several different chemicals. And now if you go down on the far left, the F2, those are the granddaughters generation. We're starting to look at early menarche and body mass index, obesity, and the shape of their bodies in their earlier life, their average age of 20. So just to remind you, the F0, the parent generation, is around 70 or 80 years old. The F1 generation is now around age 60. And the F2 generation is now an average age of about 20. And together, this forms what we call the Child Health and Development Studies. Next slide, please. So a 3-generation study that starts in pregnancy is really economical because what we're looking at is the potential for example, serum PFASs and cholesterol to impact the mother who is having her baby, the fetus, who's the child being born, the 2nd generation, and all those reproductive cells that are inside the fetus, if it's a female, or the stem cells that will form the germline in the sperm, if it's a male. And so basically 3 generations are simultaneously exposed in pregnancy. And today I'm only going to talk about PFASs, and I'm also going to mention cholesterol too in the grandmothers and their relationship to their granddaughter's body size at around the age of 20. Next slide, please. This is a quick way of telling you that what we did here is we actually measured body mass index, blood pressure, waist circumference in 2 generations of women, approximately age 50 for the F1, that's the daughters who were born into our pregnancy cohort, and at age 20, which are the granddaughters who were paired with their moms. And we're going to look at those measurements in the granddaughters in relation to the grandmother's PFAS chemicals and cholesterol during the time their grandmother was pregnant with their mother. This gets complicated pretty quickly, but I think if you keep that other picture in mind, it'll help. Next slide, please. So this is what we found. So the granddaughters whose grandmothers had lower levels of cholesterol, but higher levels of one particular PFAS compound called ETFOSA, had a significantly increased risk of both abdominal and whole body overweight and obesity. There were other findings too that were related, but this is the primary finding that I'd like you to take away from this conference. I'd like to emphasize that we don't understand the mechanism for this association, but we do know that this finding has implications for the current epidemic of obesity if this can be confirmed. Why? Well, because the grandchildren who are age 20 now and their moms who are age 50 or 60 now are part of this very large obesity epidemic. And I've already showed you that the exposure to these compounds is very widespread and started in the 1940s and 50s and is consistent with the timing of the obesity epidemic. And finally, there are implications for PFAS cleanup, if this is confirmed, and opportunity for setting up precautions for pregnant women for the major medical societies about how to try to avoid this contamination, to avoid sort of a rekindling of a 3-generation of effect 60 years down the road. Just to remind you, abdominal obesity means a waist circumference more than 88 centimeters, which is about 34.6 inches. And whole body overweight or obesity is a body mass index of greater than or equal to 25 kilograms per meter squared. Next slide, please. So I want to emphasize that these results for the granddaughters were not because of the grandmother's obesity, were not because of their own mother's obesity, were not because of the race or ethnicity of the individuals that might have been correlated with their exposure. Next slide, please. So We have to talk a little bit about ETFOSA because it's not rolling off everyone's tongue. It has not been very well studied. But this slide is a schematic, and it greatly simplifies what's going on here. There's a raw material called ETFOS that was synthetic and was used to manufacture most of the legacy PFASs in the past, and it's highly it's found in the environment, and it was a source for the manufacture of the chemicals in all of those products that we mentioned before. And then there's this stuff, ETFOSA. Well, ETFOSA is a transformation of this raw material ETFOS, and it's produced in the environment and in living things, and we can find it in the environment and people. And it isn't often measured in studies, but that does not mean it's not there. Importantly, it is a step in the transformation to PFOS, which is a chemical you will have heard a little more about. This one is a very stable forever chemical found in people and the environment. And what many of us are concerned about are all of the thousands of compounds between all these arrows, some of which, or many of which could be toxic, and which could be important in understanding the health effects of the compounds in general. Next slide, please. I just want to show you what I'm not going to make you memorize today. These are some of the arrows that are more complete picture of what's going on here. And if you look really carefully, I can't use my pointer. ETFOS is kind of on the left-hand middle of the slide. There you go. Thank you, Jenny. And the ETFOSA is formed from it, and through a series of other steps can be transformed eventually to PFOS up at the top there, Jenny, if you can find it. So this chemistry is only a very, 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 very small part of what we know about these chemicals, or what we need to know about them. There are tens of thousands of PFOS and PFAS related chemicals, and we don't know very much about them yet. Next slide, please. So just a little bit on where ETFOSA comes from. This data is very new, and it's unusual that it's there at all, and I'm very grateful for it. What we think is ETFOSA is, which is the compound we're looking at here, is in consumer products, and that there's a migration of this stuff from carpet cleaning products and food packaging materials. And I really direct you to this lovely paper by Hugh et al. The references in here to get some more information about why we think this is true. But more than one study has suggested that ETFOSA's main source is consumer products. Others of these chemicals, the main source might be water, the main source might be food, the main source might be fish. Next slide, please. So here are the takeaway messages. Previously, in our 3-generation pregnancy cohort, which is called the Child Health and Development Studies, we found that maternal serum ETFOSA in combination with cholesterol, again, cholesterol is involved somehow, either in the transport and metabolism of this stuff, but it was correlated with daughters' risk of breast cancer, which we haven't talked about today, but this work is already published. Today's talk is the new finding that we were to present at ENDO 2020, the grand maternal ETFOSA in combination with cholesterol correlates with granddaughter's obesity. I mention this because we looked at the cholesterol interaction, which was statistically significant, because we already found it in relation to daughter's breast cancer. So I just want to emphasize this was not a fishing expedition, but it was a direct hypothesis that was tested. Finally, I'd like you to remind you that ETFOSA is a metabolic byproduct of chemicals that are used to manufacture other PFASs in consumer products, such as food packaging and carpeting. And this therefore may contribute to the current epidemic of obesity should this work be confirmed. And finally, in some ways, it is at least the most important message to everyone. Pregnancy appears to be, based on our study and other animal research and other work in progress, pregnancy appears to be a critical window of exposure for at least 3 subsequent generations in humans. And next slide, please. I'd like to say a few words about the future of our cohort. This Child Health and Development Studies, CHDS, is the only existing opportunity at this time for 3 and eventually soon to be born 4th-generation research on the impact of environmental chemicals during critical windows of pregnancy and in the womb. This is very important. It would take another 60 years to start such a study now. There are other studies in progress that 20 or 30 years from now can do this, but we need to keep this data available. We are actively seeking funds to secure the cohort for the future of science, not just for research that we're doing right now, but it's to store biospecimens, to communicate with CHDS members, to enroll the great-grandchildren formally as they're born and collect biospecimens and other things. I really encourage you to contact me if you have any ideas or can help fund the infrastructure for this study. Next slide, please. We'd like to thank many, many funders over 60 years, but several critical awards, most notably our 3 Generations Breast Cancer Study Award and the Germline EDC Exposure and Breast Cancer Risk Award that came from the California the California Breast Cancer Research Program have supported this work directly. Multiple institutes over 60 years, but mainly the National Institute of Child Health and Human Development. And the state of California, which created and supports the California Cancer Registry, the Vital Status and Department of Motor Vehicle Files, without which this birth court could not exist. And finally, it really takes a village and more like a whole state and nation to do this work. And these are only some of the people involved, not to say the least, the 50,000 people that are now represented in this cohort. We thank you also for listening and for continuing to actually support this work by, if you're a Californian, to check off funding for the California Breast Cancer Research Program. And if it's a national program to the Department of Defense Army Breast Cancer Research Program. That's it. Bye.
Thank you so much, Dr. Cohn. I'd now like to introduce Dr. Mohan Kumar. Oh, I'm sorry, you're on mute. Hold on one second.
Can you hear me now?
Dr. Mohan Kumar.
Yeah, okay. Good afternoon, good morning, good evening, depending upon wherever you are. Thank you, Dr. Cohn, for that excellent talk. So this kind of follows a little bit of what you have done, but in a totally different set of chemicals. So these are going to be bisphenols, and you are probably, all of you probably know about bisphenol A. That's the most well-known compound. But then instead of bisphenol A, after knowing all the bad effects that it might have, people have started using bisphenol S and bisphenol F. So our goal was to see whether these are safe, are as good as bisphenol A, or worse or not. That was the main goal of this project. Next slide, please, Jenny. So my focus is going to be in this talk, particularly is going to be on cardiovascular diseases, even though we have looked at the effects of this exposure, prenatal exposure to these compounds on various other systems, such as the reproductive system and the metabolic system and things of that nature. I'm going to be focusing on the cardiovascular function. So as you all know, cardiovascular diseases is one of the leading causes of mortality among men and women, and a significant population of the United States is believed to be hypertensive. And there are multiple reasons, various reasons that you can attribute to for this increase in prevalence of hypertension. But one of the things that has not been studied well is the exposure to environmental chemicals during pregnancy. So that's what we are going to be focusing on. Next slide, please. So there's a little bit of a background about bisphenol. So like I said before, BPA is a widely used agent that has been known to contaminate the environment. And we are exposed to BPA and related compounds on a regular basis, daily basis. And the important thing is BPA and related compounds easily cross the placental barrier. So what do we mean by that is, so the placenta creates a barrier and prevents unwanted compounds from getting into the fetus, obviously for good reasons, not to affect the development of the fetus. Whereas BPA and its related compounds are known to cross the placental barrier easily, and they can influence the development of the fetus. So As I mentioned before, currently BPA analogs such as BPS and BPF are used a lot in the plastic industry, even though they are not high volume compounds like BPA, but they are slowly replacing BPA. So our focus was to find out what is the effect of these compounds, particularly BPS and BPF, in terms of how it affects the development of the fetus when moms are exposed to it during pregnancy, and then follow it up after the fetuses or the female offspring reach the adulthood, then follow what happens to their blood pressure. So that is the main objective of this study. Next slide, please. So we hypothesized that the prenatal exposure to BPA, BPS, or BPF will increase blood pressure in female offspring. So to investigate this, we used an animal study. Next slide, please. So in this study, we used spread dolly rats. These are female rats, and they were made pregnant. And during the days of the pregnancy in spread dolly rats, it's about 21 days. So we exposed these moms either to the vehicle for these compounds or BPA, BPS, or BPF at the doses that are shown there on the slide, 5 microgram, 5 microgram, or 1 microgram, respectively, during the days of pregnancy of 6 to 21, okay? And you can already see that instead of 5 microgram in BPF, we have only 1 microgram. You would be wondering why we have just 1 when we have the other 2 compounds as 5 microgram. The reason is we tried with the BPF at 5 microgram and we found out that lots of animals aborted. So already BPF 5 microgram dose was pretty not good. So we had to reduce it down to 1 microgram. At this dose, the animals did not abort, but then you will see what happens to the cardiovascular system very soon. So the animals, developed and they were born, and right around they're about 3 months of age or 3 to 4 months of age. These animals were implanted with a radiotelemeter. So a tether was implanted in femoral artery, and that helps us monitor the blood pressure without touching the animal. Second by second, you can monitor it if you want to. Systolic blood pressure, diastolic blood pressure, activity, all those things can be monitored using these devices. So what we did in this study was we recorded these measures specifically over a 24-hour period at 10-minute intervals and every week. So in the next few slides, I'm going to be showing you some of the results from this study. Next slide, please. Next slide, please. Okay, so this slide, as you can see, we are showing you the blood pressure data. So the one on the left of your screen is going to be daytime systolic blood pressure. On the right side is going to be nighttime systolic blood pressure. And you can clearly see those animals that were treated, those moms that were exposed to BPA and related compounds, their offspring, female offspring, had significantly higher systolic blood pressure. It doesn't matter whether it is daytime or nighttime. So you can see the increase was the highest with BPA, but then BPS was not a slouch either. It was pretty much same as BPA, and BPF was also increasing the blood pressure levels. So can you go to the next slide, please? So this slide shows the diastolic blood pressure. Very similar to systolic, you can also see BPA and related compounds, those moms that were exposed to this compound, the offspring, the female offspring from these moms, had significantly higher diastolic blood pressure. Again, it did not matter whether we measured them during the day or night, it was significantly higher. Next slide, please. So very similar observation in terms of mean arterial pressure. Next slide, please. So overall, what I can summarize here is when moms are exposed to the doses that we have talked about, which is 5 microgram BPA, 5 microgram BPS, or 1 microgram BPF, the female offspring that were born to these moms had elevated systolic as well as diastolic blood pressure. And this increase was at least 20 to 25 milligrams of mercury higher than the control animals. So that is pretty significant. So not only these animals are hypertensive, or even if you want to think that they are in pre-hypertensive state, so keeping these animals at pre-hypertensive stage for such a long period of time, you're going to have lots of cardiovascular issues later on. Next slide, please. So in conclusion, we can clearly see that prenatal exposure to bisphenols or the related compounds definitely increase the systolic and diastolic blood pressure in the offspring. And the doses that we used in the study, I just want you to note that they are less than the EPA recommended exposure doses. At present, we do not know the mechanism by which this exposure causes the blood pressure increase. But based on our prior studies, we suspect several mechanisms that could be happening in the brain. We think that it might be a central mediated phenomenon, but we do not have concrete answers to those questions yet. We are looking at the mechanism right now in our lab. Thank you very much.
Thank you, Dr. Mohan Kumar. We're ready for questions and answers, and I see we already have some for Dr. Cohn. So Dr. Cohn, this is from Jim Kling with Clinical Endocrinology News. Was there any effort to measure the presence of the chemicals in daughters and granddaughters, or to look at epigenetic changes that may have occurred as a result of exposure? Oh, I'm sorry, you're still on mute.
Oh, good. That's a great question. So the answer to that is that we haven't done that yet. We really would like to do it. We have done this once already, looking at the DDTs. So we have looked at in utero DDT exposure in relation to the epigenome at age 50 in the daughter's generation, and we have published a paper on that recently in Reproductive Toxicology. It's actually very interesting. There are a number of breast cancer related genes that do appear to have changes in their, at least in their DNA methylation. So we would very much like to continue this work and do exactly what you suggest, because we understand that there may be multiple hits occurring across these generations at very important times. And first, we would like to see whether we can find any epigenetic changes that might be shared daughter to mother first, and then in the fourth generation, whether they will be maintained. In addition, each of these generations have had different hits at different points in time. We're very fortunate that we're going to be able to recover some of that someday. California stores blood spots, and so the births of these granddaughters have exposures recorded at least at the very important newborn period that we could get from these blood spots if we can ever find the funding to link them. So what we're aware of is that there are probably likely multiple hits down the line, and we're not really sure how they all match up. It will be important to understand that. Also because the exposures are changing themselves. We already have documented that. So as we migrated away from the C8 compounds to the C10 compounds, what you see in the blood, for example, of the grandmothers at age 30 doesn't look exactly like what you see in the blood of their daughters at age 50. Too long of an answer, but we're working on it. Good question.
Thank you. I've got two questions from Elizabeth Havlinka at MedPage Today for Dr. Cohn. One, do you have a hypothesis related to the mechanism by which these compounds are impacting generations of women? Do you think it is related to your findings on PFAS and breast density breast cancer?
That's a really good question. I guess I don't think we're ready to say how this might be happening. We do know that these compounds, the PFAS compounds, seem to disturb lipids and lipid metabolism. And so the idea that there is this actual interaction, at least a statistical interaction with maternal cholesterol and these compounds for producing or correlating with these effects in the children, in the grandchildren, and in the daughters isn't surprising. But I think it's really too much early days for us to speculate. We're working on it. I have wonderful colleagues who are toxicologists who and geneticists at UC Davis, and we're working very hard to try to understand what physiologically and biologically might be happening here. We don't know yet.
She had one follow-up question. How was exposure to ETFOSA measured?
Oh, the California Department of Toxic Substances and Control, the Cal EPA, has a fantastic laboratory. It's the laboratory of Mirto Petraeus and Judith Sue Park. It was measured in the serum of these pregnant women, And the serum we chose to use was early postpartum serum, which was collected 1 to 3 days after delivery. These compounds are very persistent, and we think they very much reflect what the fetus was exposed to. Eventually, we like to save the time samples for things that are absolutely critical that change over pregnancy. So if we do have first, second, and third trimester samples as well. At some point, we're probably going to go back and look at timing issues, but we haven't done that yet. But these are serum levels that we've looked at, not reports or estimates of any sort.
Right. I have a question for Dr. Cohn from Sherry Roan with Everyday Health. She asks, can you speak to current efforts to help people avoid PFAS exposure?
Oh, absolutely. That's doable. Well, it's doable and it's not doable. The biggest effort surrounds food packaging now. A lot of the things that are inside that cup that doesn't disintegrate, even though it's paper, the lining inside pizza boxes, some of the compounds that are used basically make things slippery and make things resistant to moisture. And so anything that you can do to reduce your exposure to those things are things like not using takeout containers, using stainless steel or glass whenever you can for all kinds of storage purposes. And it turns out that eating lots of takeout and to-go food, because it's often prepared in contact with some of these compounds, is something that can help you avoid not just this chemical, but some others that are like the BPAs that are in the plastics that the other talk mentioned. So then the stain master carpetings and all of the stain resistant and water resistant substances that you have in your house are important. The carpeting is probably extremely important because these compounds don't stay on the carpet. They are sort of emanate as dust. And the other thing that is related to that is that you can keep your house as clean as possible. You can dust as frequently as you can. If you have children in particular, this might be important. Pulling up that old carpeting and using just plain hardwood flooring and washing it regularly can help also avoid these compounds. The problem is personal measures probably are not going to be enough because like the other legacy compounds, the PCBs and the DDTs and some of the pesticides, they've been so ubiquitous, they're so much everywhere, and that they're in our food and water. You can't do 100% avoidance, but some of these other techniques can work and make you feel that you have some control of your life.
Thank you. Dr. Mohan Kumar, I have a question for you from Jim Kling from Clinical Endocrinology News. He asks, why female offspring? Also, do you know why BPS and BPF were chosen as alternatives by industry? Were they tested in some way before being used as replacements?
Okay, excellent. Thank you for the question. So even though this study that I'm talking about female offspring, we are also currently looking at the male offspring, and we are implanting telemeters and looking at the effects. So we intend to find out whether there is any gender difference in terms of offspring, whether you see difference in terms of effects. But the reason why we actually studied the female offspring first is because of our prior studies where we had shown estrogen exposure at low levels to female offspring can cause hypertension. And that's one of the reasons why we followed the female offspring first. But like I said before, we are going to be looking at the male offspring also. To answer the second part of the question, that is why industry uses BPS and BPF as alternatives. So it's not clear why they chose, but I think the rush was to declare the compounds BPA free. And when that happened, they had to find alternatives. And BPS and BPF are structurally extremely similar to BPA. So they started using BPS and BPF instead of BPA in parallel with BPA, I guess. So it's not clear why they chose those two compounds, but these two compounds have been in use for quite some time now, and you can detect them in and all sorts of human tissues and animal tissues. So that's an answer. Then the third part of the question is, were they tested in some way before being used as a replacement? That was the third part of the question. I don't think so. At least I'm not aware of the industry doing specific studies in terms of, or published studies that I know of, that they have looked at the effects of BPS and BPF and compared it with BPA. But these days, we are seeing lots of literature showing these two chemicals can be, if not worse, at least equally bad to BPA. But the present study that we were supposed to present in this endocrine meeting clearly shows for the first time that when moms are exposed to these compounds, which are so-called analogs of BPA, the female offspring from these animals can develop blood pressure. That's extremely significant. And as Dr. Cohn was talking about, typically when animals or humans are exposed during pregnancy, the effects can be generational, multi-generational, so it can go to the second generation or the third generation. Obviously, I can't say based on the current data whether these effects that we see with BPA or BPS, BPF will go to the next generation or not, because I don't have the data to show that. But it's quite possible, and we have to be aware of those possibilities.
Great, thank you so much. That's all the questions we have for right now. If anyone has any additional questions, please type those in the chat window. I would direct you, we have some new clinical resources for clinicians on understanding EDCs. It's on our website. It's a feature called Let's Talk EDCs. Feel free to email me at media@endocrine.org if you'd like a link to that. And I think that's all the questions we have. So again, thank you so much to our speakers and everyone else for joining us. We really appreciate your time and have a great day.
Thank you so much. Thank you so much. Appreciate it.
Thank you. It was fun. I hope it was helpful.